All you cool cats and kittens, welcome to the Bacterial Transformation Lab activity. I'm going to recommend that you have printed this out and have it in front of you as I go through this tutorial to explain and tell you what to do, just like if we were in class together. Okay, so if you haven't done that, do that now. Also, if you have not watched and taken your notes from the video guide for the Lab 6 Bozeman video on bacterial transformation, do that now so that you can familiarize yourself. And I did teach about this segment in my video on biotechnology the other day as well. All right, so if we're ready to begin and you have everything in front of you that you need, some background information. Agar is made from agarose, a sugar nutrient medium to grow bacteria placed on the bottom of a petri dish. Using the information provided and considering the environmental conditions of each of the 4 petri dishes in a bacterial transformation experiment, make predictions for what you would expect to find growing or not growing in each petri dish below. Remember, plasmids are small circular pieces of DNA in prokaryotes that can be used as vectors. Ampicillin is an antibiotic that kills bacteria, like E. coli. Loria broth, LB, is what the E. coli are normally grown in lab conditions before the bacteria is plated on petri dishes for experimentation. Bacterial transformation is transfer of genetic material, the plasmids, between 2 bacterial cells in direct contact. Okay, so here's your table 1, and what you're going to do is think about what you would actually see. You are going to make some predictions in the appropriate rows. So each of these 4 is going to go into a row based on what the plate is. Okay, so make your prediction there. Here's plate number 1. This is your Loria broth plate. It does not have plasmid in it. There is no plasmid in it. Plate number 2 is Loria broth, but it also has ampicillin in it, but it does not have the plasmid for the bacteria, so there's no plasmid. It's going to mean no ampicillin resistance for that reporter gene. For plate 3, this is Loria broth plate with a plasmid, and that plasmid has the ampicillin resistance reporter gene in it. Plate number 4 has the Loria broth, ampicillin in the plate, and it has the plasmid, which has the ampicillin resistance reporter gene and the P-glow gene to make it glow. Okay, so pause the video here, write your predictions below, and these are in no particular order, so try to match them up as best you can. Pause the video now. All right, once you've done that, we're going to come into the pre-lab questions here about your predictions and to see if you have a good understanding of what you think you should see. Question number 1 says, if there are any genetically transformed bacterial cells, on which plates would they most likely be located and why? So looking up at what you predicted, tell me where you think those genetically transformed bacterial cells will show up, what plate numbers, and why would they be there. Okay, you can't just put the plate numbers. You have to tell me why. All right, number 2, which plates should be compared to determine if any genetic transformation has occurred? Which plates are you comparing to what and why? Okay, so there are 4 plates here. There's a reason there's 4 plates. Tell me which ones you're comparing. What's meant by a control plate? Answer that question, and then you will also answer the question as to what is the function of each plate you set up. If you're not exactly sure, make sure you go back to your notes, go back to the video. All right, so pause the video here and answer these 3 questions completely. Okay, so obviously we didn't get to do this in class. You would have done this on as a double period. It would have taken us a few lab periods on that Monday that we were supposed to come back. So what I've prepared for you is my own drawing of what the experimental results should have been. Okay, so that is available to you, and it looks just like this. So you can print it out if you like, or you can just have it up on the screen. Here's plate number 1. Your Laurier plate has no plasmid in it. Here's plate number 2. It's got Laurier broth and ampicillin in it, no plasmid. Here's plate number 3 here, the Laurier plate with the plasmid for the ampicillin resistance. The colonies are white, so each of these represents a bacterial colony of several thousands of bacteria in each. That's why you can see them. And then over here is the Laurier broth and ampicillin plate, and it's got the plasmid with the ampicillin resistance gene in it, and also the gene to make it glow. And since I only had a blue colored pen, our colonies are glowing blue here. Okay, so you can have that out in front of you. So you're going to use these to come back here. You will write your observations for each plate here. You can make a bulleted list, is fine. And then you're going to draw what it looks like. Okay, so do your drawing over here, and don't forget to label each petri dish in the drawing below. Right, so put the number of the petri dish. Okay. And once you've done that, we're going to use your results here to answer the post-laboratory questions. Okay, so you will use your observations of what you saw on the plates, and you're going to answer questions 1 through 7 for that. Okay, then we're going to get down to number 8. I'm going to teach you how to calculate transformation efficiency. And so in order to do that, we have to count the number of colonies. So you're going to come back here. Here's our plate number 3. You will literally count these up. 1, 2, 3, 4, 5, 6, etc. Count them all up. So you will put that, right here, the number of colonies, white colonies in that petri dish. And then you will do the same for the colored colonies. Okay, so come back to your picture. Count up each of these colored colonies. There is no difference in the color, it just depended on if I was running out of ink or not. So count all of these up, the colonies that glowed blue, and then you will record the number of colored colonies right here. Okay? All right, and then we're going to calculate transformation efficiency. So I'm going to kind of take you through the steps to do so. You will need a calculator for this. If you do not have a calculator at home, go on the web, type into Google calculator, and one will pop up for you, or you may use the calculator on a phone. All right, transformation efficiency is expressed as the number of antibiotic resistant and glowing colonies per microgram of pea glow. Each colony on your Laurier broth, ampicillin, plus the plasmid that has the reporter gene and the pea glow plate can assume to be derived from a single cell. All right, so determine the total number of blue fluorescent colonies. You should have done that already. So take your number of color colonies right here and write it down right there. Okay, so that will give you the number of colonies there. Then we'll move on to step 2. You're going to determine the total amount of pea glow plasmid DNA in the bacterial cells that are actually spread on the petri dish. Okay, so you're going to get your total mass of the plasmid in the bacteria is equal to the volume of DNA used in microliters times the concentration. So come right down here. You used 10 microliters of pea glow. That's your volume of DNA. See it? At a concentration of 0.08. Multiply those. Put that answer right there. For number 3, you're going to determine the fraction of pea glow plasmid in the E. coli that you actually spread on the plate. Okay, so this is going to be equal to the number of microliters of bacteria you've spread on a plate. That is 100 microliters from the procedure divided by the total volume in the pea glow microtube. Okay, and down here it's telling you how you get the total volume. Okay, so you have the amount of calcium chloride used, that was 250 microliters, plus the amount of plasmid used in step 2. Okay, the amount of plasmid used in step 2 is right there. Okay, so you've got 250 plus whatever you figured out from here, plus the amount of Loria broth, which is another 250. 250 plus 250 is 500 plus whatever micrograms you have here. Okay, so add those together. That will give you your total volume. You will take the 100 microliters, 100 divided by the total volume you just calculated, and put that number right there. That's the fraction of piglo that you spread on your plate. For number 4, you're going to determine the mass of the piglo in the bacteria. Here's our piglo mass is equal to the total mass from step 2. Go up. Total mass in step 2 right there. Okay, times the fraction of the spread that you actually used in step 3. You will take this and you will multiply it by that, and then you will put that right down there. Okay, that's that. And then for 5, you're going to determine the number of colonies per microgram of plasmid used. The number of colonies that you counted in step 1, boom, this number right here, whatever you got, divided by the mass of the piglo plasmid from step 4, divided by that. So for 5, to get your transformants, it will be number 1 divided by number 4, and that's going to give your transformants. And that is how you calculate transformation efficiency. Okay? So you're going to use the same steps above. I just took you through that, but I want you to practice it. Okay? And you're going to calculate the transformation efficiency for the number of white colonies here. All right? So you will follow each of these steps. I want to see your work. Okay, so show all of your work so that you can get credit for that particular question. Okay? All right, so that's how you do the bacterial transformation activity. As always, if you have any questions, please hit me up with an email, or you can send me a question through the Remind, and I will be happy to help you. Have a great day, everybody.